subdivided to humoral and cellular immunity, and antibody is the main component of humoral immunity. The main topics of this class is: what is antibody, structure of antibody, features of antibody versus of other molecule involved in recognition, and finally, I speak about different types of antibody. Mr. White, are you record my voice? Mm. 1 minute. The main topic of this class is what is antibody, structure of antibody, features of antibody versus of other molecules involved in recognition, and finally, different type of antibody. As you know, immune systems divided to innate and adaptive immunity, and adaptive immunity subdivided to humoral and cellular immunity, and antibody is the main molecule in humoral immunity. Antibody is produced by activated B cells or plasma cells. An immune system use this molecule to identify and neutralizing foreign subjects such as bacteria and virus or other chemical antigens. The antibody recognizes a unique part of foreign target called an epitope. So, epitope is the specific part of antigen that antibody binds to it. A close look to general structure of antibody showed that antibody composed of four polypeptide chain, two long chain that names is heavy chain, and two small chain or light chain. This chain bind to each other with disulfide band. As you see, each chain composed of a very specific area in the globulin domain. A closer look shows immunoglobulin domain is area composed of 110 amino acid regions that all functions of antibody molecule is related to this area. There are two different types of immunoglobulin domain. One area near to the N-terminal names is variable domain, and other domains in the C-terminal named constant domain. So, light chain or small chain composed of two immunoglobulin domain, one variable domain and one constant domain, while the heavy chain or large chain composed of one variable domain and 3 to 4 constant domain. All function of antibody molecules is related to constant domain, and variable domain only bind to antigen. So, one molecule of antibody has two antigen binding sites, 1 and 2. The sites bind to epitope on the antigen surface. This figure shows binding of molecule antibody to the epitope site on the antigen surface. And subtle investigations show that every variable domain composed of 3 hypervariable regions names is CDR. So, each variable domain composed of 3 different CDR, CDR1, 2, and 3. And CDR3 is very important in antigen binding. So, if amino acid change is occur in CDR3, it's led to ineffective antigen binding, or it may be led to hyper affinity maturations. This figure shows the variability of CDR regions on a variable domain. As you see, CDR3 is very variable or highly variable. You see, variability, residue position number, immunoglobulin domain composed of 110 amino acids, and CDR3 has hypervariability in comparison to other CDR. So, immunoglobulin domain are area that contain 2 layers of beta sheet, and each layer composed of 3 to 5 strands. 3, you see with different color. 3, 1, 2, 3, 1, 2, 3, 4, 5 strands. So, immunoglobulin domain contain of two beta sheet layer, and each layer composed of 3 to 5 strand, anti-parallel strand. In immune system, there are three molecules that participate in antigen processing and antigen recognition. One is immunoglobulin or BCR or B cell receptor, other is T cell receptor, and finally MHC molecules. In the next class, we want more detail. I speak more detail about MHC molecules. These molecules are different in the nature of antigen that may be bound, nature of antigenic determinant recognized, and affinity maturation. So, antibody bind to different molecules, protein, lipid, polysaccharide, and a small chemical component, while TCR and MHC only bind to the protein or peptide. Antigenic determinants are linear or conformational. Antibody bind to linear and conformational determinant, while TCR and MHC only bind to linear determinants. And antibody, we can see affinity maturation about antibody or immunoglobulin, while there is no affinity maturation about TCR and MHC molecules. A healthy person produces about 2-3 grams of antibody every day. Almost 2/3 of this antibody called IgA. More detail show that there are two different types of light change, kappa or lambda. We see two different types of light change in antibody molecules, kappa or lambda. In human, about 60% of antibody molecules have kappa light change, and about 40% have lambda change. This ratio is very important in medical medicine. Marked change in this ratio can occur in patients with monoclonal B-cell tumor because the neoplastic clone produces antibody molecule with the same light change. In fact, the ratio of kappa bearing cell to lambda bearing cell is often used clinically in the diagnosis of B-cell lymphoma. There are 5 different types of heavy change: lambda, mu, delta, epsilon, gamma. When antibody molecules have lambda change or alpha change, this antibody named is IgA, mu, IgM, delta, IgD, epsilon, IgE, and gamma, IgG. So, the name is antibody molecules completely depend to heavy change, not light change. When heavy change is mu, antibody names is IgM. A closer look to antibody structure showed there are 3 different determinants: isotypic determinant, allotypic determinant, and ideotypic determinant. Isotypic determinants located completely on constant region of heavy and light change. And allotypic determinants were scattered in constant region. On the light change and heavy change. And finally, isotypic determinants were located on variable region or variable immunoglobulin domain. So, three different determinants were located on different location of antibody molecules. Depends to heavy change, we can see 5 different class of antibody: IgM, IgG, IgD, IgA, and finally IgE. 2 types of this class, IgG and IgA, divided to different subclass. IgG divided to 4 subclass: IgG 1, 2, 3, and 4, and IgA subdivided to IgA1 and IgA2. In this figure, we can see different aspects of different types of antibody molecules. Heavy change, serum concentration, serum half-life, secreted form, and function. This antibody have different function and different location in the body. For example, IgA participate in mucosal immunity and IgE in defense against parasite hypersensitivity. All 5 class of antibody on the B cell surface has monomeric form, but IgM and IgA in secreted form has pentameric form about IgM and dimeric form in IgA. Comparison the serum concentration showed that IgG has more concentration in comparison to other class, and about serum half-life showed that IgE has the minimum half-life, 2 days. Comparison the carbohydrate concentration showed that IgD and IgM have high amount of carbohydrates. In this figure, you can see IgM structures. The main feature of IgM is low affinity and high avidity. Affinity is the connection power of one antigen binding site to its epitope, and avidity is related to all antigen binding sites. So, affinity, one antigen binding site, and avidity, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 antigen binding site. The connection power is avidity. The main feature of IgA is protection of mucus. First, IgG. Near to 80% of total antibody in the serum is IgG. And this IgG is only immunoglobulin to cross the placenta in medicine. More details show there are 4 different types of subclass about IgG: IgG1, IgG2, IgG3, and IgG4. And IgG3 has half-life near to 7 days. Other form has half-life about 2-3 or 23 days half-life. IgM is the first antibody to be produced in a primary immune response. So in the first antigenic challenge, B cell produce IgM, and this molecule is very efficient in complement fixation. In the IgM structure, you can see light chain, heavy chain, and J chain. This chain is produced by B lymphocytes. Light chain, heavy chain, and J chain. The next class is IgA. IgA is the second most abundant immunoglobulin in serum, while in total body, IgA has a high concentration. IgA is present in the breast milk, tears, mucus, and gastrointestinal tract. IgM in the serum is primarily in a monomeric form, but in secretion form, dimeric form is predominantly. In the antibody structure, you can see light chain, heavy chain, G chain, and secretory component. Secretory component is not produced by B cells. It's produced by epithelial cells. You can see after antigenic challenge, activated B cell or plasma cells produce IgA, and this IgA binds to poly-IgA receptor on the epithelial cell surface, and this combination, after enzymatic cleavage, some part of this receptor remain with antibody structures that name is secretory form. The main function of secretory form is the protection of IgA structure about microbial enzyme. You see, lumen, epithelial cell. In the lumen, there are so many bacteria that many of these bacteria may be pathogenic form, produce different type of enzyme. And secretory chain you know, secretory component protects IgA structure about this enzyme. As mentioned previously, IgA subdivided to two different types, IgA1 and IgA2. In serum, 90% of IgA is IgA1, and there is no allotype in this form of antibody. And IgA2, the main feature is there is no disulfide bond between heavy and light chain. And in secretory form, IgA1 and IgA2 has the same concentration. In serum, 90% is IgA1, but in secretory fluid, IgA1 and IgA2 has same concentration. And IgD mainly at the antigenic receptor on B cells that have not been exposed to antigenic. You know, lymphocytes divided to naive lymphocyte, activated lymphocyte, and memory lymphocyte. In the naive lymphocyte, IgD present as the BCR or B cell receptors. And finally, IgE. IgE allergens and trigger histamine release from mast cell and basophil and is involved in allergy. Also, IgE is very important in protect against parasites. We can also divide antibody to different types: monoclonal antibody and polyclonal antibody. Notice there are 5 different types of class antibody, and we also can divide all of this antibody to 2 different types, monoclonal and polyclonal. Monoclonal antibody is a form of antibody that produced by only one B cell clone, and it's produced against one epitope. And this form of antibody is very important in medicine. Monoclonal antibody. So, monoclonal antibody is a form of antibody that produced by only one B cell clone. But polyclonal antibody is a form of antibody produced by multi-lineage of clones, and it's produced against different type of epitope. For example, if a snake belongs against a snake venom, we can use polyclonal antibody for protection. So, or you have any question? Do you have any question? In the next class, I speak about genetic of immunoglobulin, and I think it's enough for you. If you have no questions, the class is finished. You're welcome.